I'm going to show images of germ cell tumors of the central nervous system. They have a peak incidence in adolescents between 10 and 15 years, and the majority of germ cell tumors in the central nervous system are germinomas. Germ cell tumors occur in the midline, and in the central nervous system the most common location is the pineal region, followed by the pituitary region, and sometimes germ cell tumors are bifocal, as in this case. They have relatively low T2 signal and heterogeneous contrast enhancement. As the name says, they are derived from germ cells, and germ cells do something quite peculiar during embryology. The germ cells migrate out of the embryo during gastrulation. They divide and then move back into the embryo. And it has always puzzled me why these cells would do that. I got married 15 years ago, and when I got my wedding pictures, about 3 weeks after our wedding, I realized I was doing the same with my wedding pictures as the embryo does with the germ cells. I'm going to talk about my wedding pictures first. So I got my wedding pictures 3 weeks after our wedding. I immediately made copies of them and gave them to my parents and my in-laws, just to be sure that even if our house would burn down, my wedding pictures would be somewhere. And in our house I stored them in an iron box in our living room. And the house we lived in the first 10 years of our marriage was near a river. And in times of high water level, I would relocate the wedding pictures from our living room to our bedroom on the first floor. The embryo does the same with germ cells. So during gastrulation, when the embryo goes from a bilaminar to a trilaminar disc, it knows it's going to survive and that it can set aside some cells for the next generation. So from each region of this embryo, cells are being set aside and they migrate near the hindgut yolk sac junction. And in this Carnegie stage 9 embryo, this region has been outlined in green. And these are other pictures of a gastrulating human embryo where you can see the embryonic disc, and this is the region where the germ cells are going to be. So this is gastrulation, just after the blastocyst has implanted into the endometrium, but there's no placenta yet, and there are no blood vessels. And these cells are really important for the embryo, like my wedding pictures were for me. And the embryo wants to keep these germ cells safe. So it saves them and it keeps them in the region where there are a lot of nutrients. So all the glucose has to diffuse into this embryo and all the nutrients are in the yolk sac. So if you keep the cells near the yolk sac, it's where you're sure that they will have enough nutrients to survive. Then the embryo develops further, so it folds and it becomes the form of an embryo. And then the primordial germ cells or stem cells migrate back into the hindgut, and then along the hindgut, along the midline, and then they move in the dorsal mesentery in the genital ridge, and this is going to become the gonads. And if they migrate along the midline and they take the wrong turn and they are lost and they end up somewhere where they shouldn't be, they can give rise to a germinoma. So germinomas are thought to arise from primordial germ cells. And this is an example of a 17-year-old female presenting with endocrinological symptoms and headache. And you can see a mass with low T2 signal in both the pituitary and another mass in the pineal region with enhancement. And this turned out to be a germinoma on biopsy. And she was treated with chemotherapy and irradiation. And germinomas have a pretty good response to therapy compared to non-germinomas. And this patient showed no signs of disease 9 months after treatment. As shown in the previous brain bit by bit on craniopharyngiomas, germ cell tumors arise from the recess, and they can interfere with the function of the hypothalamus and the pituitary. So they might present with diabetes insipidus and an absent posterior pituitary bright spot. And on the post-contrast images of this patient with a germ cell tumor, you can see that the enhancement extends too far posterior. This is too dorsal. It shouldn't enhance all the way up to the mammillary bodies. So there was a very small germ cell tumor in this patient that was confirmed during surgery about a year later. Non-germinomas have often elevated tumor markers such as alpha-fetoprotein in yolk sac tumors and teratomas with malignant transformation, or an increase in HCG in a choriocarcinoma. And the non-germinomas do not only have low T2 signal and heterogeneous contrast enhancement, they can also be bifocal as in this 13-year-old boy, but they also have hypointense signal on gradient or susceptibility weighted images, which can be used to distinguish them from germinomas. And if you think of the teratoma with the low T2 signal being caused and the susceptibility being caused by calcifications, you know that on CT these teratomas have fatty components, calcium, and soft tissue components. And teratomas with malignant transformation, so the non-germinomas, they form a spectrum with the immature teratomas and the more common mature teratomas. And teratomas have all three embryonic layers, so endoderm, mesoderm, and ectoderm. And we're going to talk about teratomas in the next brain bit by bit.